Yeah, so all enrolled candidates, if mail ID is not shared, please share it. So post completion of this session, we're going to share the recordings of the session and the PPT in the emails. So you can download, you can save in your system as well. Yeah, so thank you all for your confirmation of enrollment and going to discuss on the especially biologics, biosimilars. Under the biologics, we can cover vaccines as well. The regulatory strategies for CMC and as well as filing pathways. So today is a introduction. So why we want to take up these sessions for 3 months? So we'll get a conclusion. So based on the tomorrow onwards, actually we can start the requirement activities for the filing activities. Yeah, is it clear to everybody?
Yes, sir.
Yeah.
Yes.
So how the filing activities is linked with the technical sessions. So we know that last 5 sessions we conducted, especially for the demo sessions. So we have been given the sessions related to the R&D development, then related to the methods which is using for testing of our analytical part, and as well as related to the process validation, and especially for the Indian market, how we are going to file the products in the different stages, and followed by variation filing. Once product get approved on the market, how we can changes our process, we're taking a permission from agencies. This is how we have been discussed. Yeah, so with respect to if you ask me how we had to be initiate the process, so even the demonstrations I given a overview, but still we require to understand at different stages different activities of regulatory activities. So just imagine discovery phase is nothing but R&D activity where we are going to research, develop the process, and going to optimize. So going for the technology transfer. So prior to that, when you want to initiate any biological molecules development or research, so required a agency's permissions. So that is what in the day 4 session we have been discussed about IBSC role, RCGM role, and CDSO role, and the licensing activities already given a overview. In coming days, also we are going to discuss. So wherever is going to discuss the respective regulatory activities, the filing pathways will be in discussed very clearly. So in that session, I explained that especially the chemistry manufacturing controls the data which is required from R&D. So that should be understand first. Without data, so without the data, we can't go for any filing activities. So when you go to open the filing for the any checklist filling, so there is a requirement to enclose the annexures. So for example, you want to claim shelf life. So there is a different stage of stability is required with respect to drug substance, with respect to drug product. So what type of stability is required to generate for the liquid sterile injections, or it may be lyophilized powders, or it may be some other form of container closed system. So what type of data required to generate it? So those are all CMC related activities required to generate from R&D. So since we are talking about biological molecules, so from the R&D point of view, so especially upstream process development, and before that cell engineering. So how to consider the cell banks, how to categorize these cell banks, how many tiering system required in the industry expectations. So what is the benefit of two tiering system, and downstream chromatography reagents, TFF, tangential flow filtration, membranes. So how we are going to make a sterile productivity activity in the downstream stage. Similarly, drug product stage, formulation characterizations, formulation development, and filter validations. So there are many activities, CMC activities will come here under drug product case. Similarly, analytical method developments. So with respect to biological activities, it may be physicochemical activities. So what are the methods you are going to use in-house developed methods required to develop it and required to qualify it, and the qualified methods has to be transferred to the QC. So this is what we given a, even though in the demo sessions, but however it is required to understand why should discuss about this. There is a regulatory requirement. So part of our submissions, so these are all data from R&D, we have to submit it. When we have to submit, so prior to initiation of R&D, so we'll give the tentative information. So the tentative information means so and so product we are going to develop. Let's imagine we are going to develop insulin products, or the insulin analogs, or maybe monoclonal antibodies, or maybe vaccines. So we are going to propose. So this is how we are going to develop a biological molecule, or it is a biosimilar molecule. So this is the indications. Indication means the purpose of disease, especially the drug product is going to treat for the particular diseases. That is called as a indication. So we are developing this so product. So there is a drug demand in the market, or there is a patients are require this type of molecules for to save their life. So we are going to propose the regulatory authority. So they will give permission to start research. Once we start research, so we'll develop the data, and prior to conducting a preclinical trials to understand the molecule or biological molecule, whichever is developed here, where it is a safe, we want to understand. So for that reason, we want to conduct preclinical studies on the animals for understanding of immunogenicity point of view, any those toxicity point of view. To understand, we'll conduct the animal studies. So directly we can't conduct. So what is the requirement here? Again, the data whatever generated from R&D will be fill in the application for the preclinical trial application protocol preparation and brochure. So everything will be placed in the regulatory body. So the regulatory body will be review and give permission to carry out preclinical trials in the respect to approved contract research organizations who having a animal studies. So once they given permission based on the approved protocol and the drug product which is generated at R&D and the respect to agreements between sponsor organization versus the CROs will get the mutual agreement and will hand out everything to them. So they will start preclinical studies on the animals and they will generate the reports what is mentioned in the against protocol. So they will generate the data and so they will send the reports. The reports has been reviewed by our clinical team. So who is located in the our organization. So the clinical team will take care entire preclinical and clinical activities. So respect to preclinical, they will review and give the clearance for the signing off. So that signed copies will be reaching to the regulatory affairs team. So regulatory affairs team also clinical is a part of them. So they'll be jointly work it. So the regulatory affairs team will go for submission of the preclinical reports. So to the agency. So agency will see against protocol versus the report. The reports are inline. So they will approve the preclinical reports. So once it is approved, they will give a recommendation letter for to start clinical trials. So especially in India, IBSC board as well as RCGM in case of recombinant molecules, they are involving especially for the developmental activity as well as preclinical until preclinical. So let's imagine we developed the process in India market and we are looking for the developing the from India. So we have been generated until preclinical and we got the recommendation letter from RCGM. Now here onwards the decision taking by our organization, either we want to file this product in the India market or we want to file this product in the any ROW markets or want to file it to the any ICH regions or any developed markets. When you're talking about ICH regions, we can consider US FDA, EMA, Japan, and regulated markets like MHRA, Health Canada, or some other regulated markets we can consider it. So if you want to file those countries, what is the strategy for clinical trials? So this is very huge activity. Just imagine the cost wise development of R&D, just imagine 10 crores and a preclinical activity, maybe 10 crores. When you come into the clinical trials, it may be costing based on the type of region, based on the type of clinical trials we are going. It may be 100 crores, 200 crores, or maybe more than that also. So the investment will be huge here. So after investment of this much budget by the sponsor organization, if product is not approved in the US or not approved in the EMA, the entire trials, entire budget of the investment will be go wrong. So that the organization when taking a decision to file the product into the ICH region countries or any regions, so they'll be very strategic. So how will go for strategic? Just imagine marketing authorization application or with respect to we are planning for the EMA region. So prior to taking a clinical trials, so we'll go for IMPD application, investigational medicinal products. So to conduct investigational medicinal products, so what are the available data of R&D and including the biosimilarity and respect to preclinical reports we'll place here. So the moment we place here before conducting clinical trials, the EMA board, the agency, scientific advice meeting also possible. So they'll be conduct and they will understand. So what is the requirement? So they'll be review the protocols and they will be approved for the go for clinical trials. So after getting a permission from them in the IMPD meeting, or we can call it the scientific advice meetings, so we'll get the permission to carry out clinical trials in their region. So the selection of the people or it may be global clinical trial or specific to the EMA region. So we'll plan for the clinical trials. In case of new biologic, so required to conduct phase 1 first and understand the phase 1 outcome, then required to conduct phase 2, then outcome of the phase 2, we can go for the phase 3. So one by one, we'll go ahead. In case of biosimilars, since already developed this molecule, so that we'll go for directly phase 1 and jumping into the phase 3, and there is a waiver of. So based on the type of country, so there is a waiver of. In India also, the biosimilar guidelines going to revise. So there will be a waiver of will be come to the preclinical studies point of view, and there is a phase 3 point of view. And once the clinical trials with respect to stages has been conducted, so respect to batches which we are going to take the material from clinical batches, that should be generated from our clinical campaign, or you can call as a GMP batches. So these clinical trial batches, CMC point of view, the batches can be taken equal to marketing size, or we can take at pilot scale also. There is not necessary to take exactly equal to marketing size batches, or there is not mandatory to take at pilot scale also. So this decision of the taking batches, size of the batches, is ultimately decision taking by sponsor organization. The sponsor organization is taking a decision to make the equal size of the marketing batches and conducting all type of validations and studies, so that prior to going for marketing authorization, everything will be in place, so that there is a shelf life point of view, no concerns. The reason is, just imagine today 2025, in the June month we are taking a batches with respect to DS, with respect to DP, batches has been taken and placed for the stability equal to the commercial batch size, and those batches also validated and everything is in place. And the moment studies ongoing phase 1, if applicable for the phase 2 and phase 3, maybe take 1 year, 2 years, 3 years of time. By that time, our productivity will complete 2 years, 3 years shelf life. So when you placing here marketing authorization, the authority will look into deeply with the stability point of view, other studies point of view, validation point of view, everything the CMC data will be verified, including method validations and the certificate of analysis, everything will be verified, and agency will be approval for the marketing authorization. Before marketing authorization, they will come for the planned GMP, and planned GMP also will be completed. So once this is getting approved, we'll go for commercial manufacturing. So we'll get the directly approval for the commercial product. Yeah, please.
Here I got one question. Yeah. So
yeah,
in phase 1, so as you told, you can take one batch also, you can take 3 batches also, right?
Exactly.
So what is the requirement of stability? Whether you need to submit 3 batch stability data or 1 batch stability data from phase 1 is sufficient?
If validation is not conducted, validation is not conducted, 1 batch also enough. We can place 1 batch also. But
so that stability data,
1 batch also we can place.
So that stability data is enough to keep for this.
For marketing authorization filing point of view, it is for the part of your clinical data submission, it is enough. But again, you have to conduct in the large scale. So when you're going for the commercial scale of manufacturing.
So PC stage you have the 3 batches. Yeah. PC stage you have 3 batches of stability data. Then phase 1 you have 1 batch. That's enough, right?
Yes, enough.
Yes. Thank you.
Yeah. So once we got the approval, we'll start commercial manufacturing. Then we'll, if require stability, this and all, the validation is not yet completed here, then required to conduct again here. If the clinical batches with respect to volume, the size of the batches and stability, everything is same, there is no specification differences, there is no changes in the method validation point of view, no change in the plant facilities, equipments, everything, then we can execute directly commercial batches. If the facility differentiation or any equipment changes, any process changes, so after getting marketing authorization, we can take additional validation batches. So those batches has to keep stability for minimum 6 months. Then we can launch the product by comparing the previous versions. So we are talking about biosimilars especially. So biosimilars development, how we have to be choose. So respect to the biosimilarity guidelines, we have to refer to particular country. So when you're looking to the US, so refer to the specific guidelines with the country specific. When you look into the India, so definitely we'll be referring to the Indian guidelines, or we can refer ICH regions where the product is get approved in the ICH regions. We can adapt that guidelines. We can refer that one also. So that is the mandatory. Here the biosimilarity assessment, how we have to conduct R&D batches has been taken preclinical batches, which is the optimized process. So here the strategy will be 3 batches will be taken up of drug product batches versus innovator product 3 batches. So we will take up 3 plus 3 studies. And when you're going to take either one batch or maybe 3 batches or more than batches in the clinical trial batches, or it is a validation batches. So anyhow, it will be 3 batches will be there. So here also 3 batches of sponsor organization batches plus innovator product 3 batches, we will conduct biosimilarity assessment. So we have to prove that analytical similarity assessment between innovator product versus biosimilar product. And prior to marketing authorization, the expectation from agency minimum 6 batches is required. So that is how guideline has been telling. So we have to develop accordingly our development strategy. So I'll take up into the guideline references so we can see different countries what they're saying. So we can understand. Yeah, so this is our Indian guideline at present, valid guideline. Is screen is visible, the PDF copy?
Yes.
Yeah, so this is the Indian guideline. So which we are talking about guidelines on the similar biologics. So regulatory requirements for the marketing authorization. So if we want to develop Indian market, we should be refer these guidelines, which is uploaded on to the 16. Today this guideline is on under draft stage. So the industry experts are reviewed and they are providing their comments if anything. So once it is addressed and able to justify, so this guideline is going to revise, and there are a lot of changes with preclinical studies, and there is a clinical stage 3 phase 3 will be going to revise. And respect to analytical similarity, there is nothing changes. So what are the today? It will be remain same. There is no any compromise with the quality. So this guideline, what it says that, so the development principles. So especially the introduction, background, and applicable regulation guidelines and competent authorities. This is all as a introduction part. So when you're talking about especially principles, so what are the principles for the development of the biological biosimilar product? So referring a innovator product, selection of the reference product. The Indian guideline says that select the reference product with Indian market which is approved. So which is already being selling in the Indian market. The reason is the biological product is already approved in the India market, already being used by people. So that you have to use the Indian market as approved product as a reference product. So this is the one reason. And another thing, it should be approved in US, approved in the EMA, or approved in the Japan, ICH regions also we can take up, or any regulated markets also, MHRA also can be considered. Then entire manufacturing process. So respect to upstream, downstream, cell banking systems, formulations, and related to the quality, related to the analytical part, everything from R&D and scale of commercial manufacturing should be in place. And quality based consideration of similar biologics. So quality comparison, another thing, quality comparability, nothing but biosimilarity. When you take up into the biosimilarity point of view, so it is a 3 parts. One is analytical similarity, preclinical similarity, and clinical similarity. So how you have been assessed the similarity assessment with respect to innovative product versus the sponsor organization product of biosimilar. Similarly, preclinical stage and clinical stage also. So there is a dividing the animals or dividing the population. So going to treat with equally in the innovative product versus or sponsor organization biosimilar product. And we have to compare the side by side comparison. That data has to be placed here. Entire biosimilar assessment required to place here. So with respect to data requirement for preclinical studies. So what is required here? So conducting preclinical studies require pharmacodynamics, toxicological studies, and immune response on the animals. Immunogenicity has to be conducted. And data requirement for clinical application. Once we want to try for the clinical trial application in the Indian market. So these are the planning activities to be conducted on the humans. So pharmacokinetics, PD, and any confirmatory safety and efficacy data, and safety and immunogenicity data, and any extrapolation, any efficacy data. This type of things is required for the marketing authorization. Preclinical studies followed by the once it is all done, preclinical and clinical, we'll go for marketing authorization. So this is how guideline is expectation from the index part. So the same thing when you go for inside, we can see the expectation of the guidelines. So accordingly, we have to be develop the data with respect to quality, safety, efficacy. So then we are referring this guideline. Accordingly, we generate the data of CMC, preclinical and clinical. So we can get easily approval in the market from the Indian market. So agency is raising a question, anything. when we are in line with this guideline, so definitely we can able to justify by referring this guideline. When we are not followed this guideline and developed with some other strategies and we filed for the product in the Indian market, if Indian market is raising a queries with us, the agency is raising a query with us, we may be not able to answer it. So that time our product may be under hold or maybe decision making, either approve or reject. So that we don't want it. So when our marketing strategy is very clear to go for India market, we should be refer very clearly Indian market. Otherwise, we can refer any ICH regions approved product and guidelines so that we can able to justify it. Similarly, very simple guideline. So is saying for EMA. So especially for the guidelines on similar biologics. So guidelines on similar biologics medicinal products. So what is expectation here? Again, so if you see regulatory framework, scope, application for the biosimilar approach, choice of the reference product, and principle of the establishing the biosimilarity. So anyhow, in EMA point of view, preclinical will be where will be there, and clinical also many studies will be where will be there. And coming to directly, especially for the biosimilar products, it will be work on the directly biosimilarity assessment. So here the regulatory framework, what is required here? So as per the EMA guidelines, a company means our organization, the sponsor organization of the biosimilar manufacturer, choose to develop biological medicines. Product claimed to be similar, which is called as a to be similar as a biosimilar, as a reference medicinal product, which has been granted marketing authorization in the European Economic Area. So this is the expectation here. So accordingly to be developed. So when you want to compare the biosimilar assessment, so we have to procure the product from approved in the EMA market. Got this point, everybody? Yeah, is it clear to everybody? Yeah. So when you decided to EMA market, we should look into the respective country specific guideline. So accordingly, we can choose our reference product and we can buy it and we can start the development. Then complete those year in accordance with the provisions, Article 8 directive. So and so the numbering is given for the registration numberings. So according to this filing activities, what is required? So accordingly, we have to be develop our dossier development for the marketing authorization. Anyhow, that is a part of our course. We can come to know slowly how to develop the dossiers. And especially for the scope, when we are talking about, especially for the biologics drugs, especially in the EMA. So this is the agency. So Committee for Medicinal Products for Human Use, which is generally called as a CHOM. So under this, there are 4 pathways: centralized, decentralized, and there is a mutual recognitions and national procedures. So most of the biologics will go with all the biologics should be filed through centralized procedure. So there are 27 countries in the EMA region, plus there are trade-free countries of 3 countries. So when you're talking about this 30 countries you want to file, we have to go single filing with CHOM. So here will be file our EMA registration planning and will be review process will be done. That will be coming to slowly by coming days. The main thing is to file the product in the EMA. So what type of data to be generated? So as usual, R&D development, when talking about R&D, by using a quality data, nothing but by using quality by design concept, we have to generate all development activities with respect to process, with respect to analytical and formulations. When you're talking about a process, it includes cell banks, upstream and downstream formulations. When you're talking about analytical, it will cover physical, chemical, biological, microbiological, everything, so that the complete quality set of data to be developed at R&D stage and to be compared with innovator product. So we'll develop comparable product in the R&D for the especially analytical point of view. Once analytical point of view is proven, it is a highly similar, comparable, or identical. In case of biologics, to mention identical, it is a challenge. The reason is variability will be there. Batch to batch variation will come, but within the acceptance criteria, we can't call it as a identical. So the reason is because of it is a complex molecules. So we can't call as a identical. We can call as a comparable or similar biologics. So then legal basis. So the products which is already registered in the EMA or maybe US or maybe India, so we'll have a patent. So until patent expiry, we can't get approvals. So we can develop that biological product as a biosimilar, which is already approved in the so regions. So we can initiate development. We can go for preclinical. We can go for clinical, and we can submit the marketing authorization. And agency can be reviewed and can be approved but can't give the marketing authorization. So until, especially for the biological products in the US, when you're giving a 2 years is the patent exclusivity. So that today, 2025, June, we developed a approved one product. So against that, we are going to develop a biosimilar product. So anyhow, it will take 8 to 10 years time for the time period for development. So if even though 8 years we completed complete development and submission of the filing and plan GMP, everything is completed, but we have to wait for the patent expiry. And 2037 on June, when it is 2 years is going to complete so and so biological product, so we will get the marketing authorization. So the next day onwards, we can go for it. So when we are submitting a biosimilar products, there will be competition. So competition between different countries, different sponsors. So who get first? So they will get approval first. So the product which is approved first, they'll get the marketing authorization first. So it may be 1 year or 1.5 years. The third person will be not able to enter in the market unless the second person get patent exclusivity another 1 year, 1.5 years. So that will be applicable everywhere. So that legal point of view also to be considered. And general principles application for the biosimilar approach. So this is how we are going to discuss in detail coming sessions. So complete biosimilarity, expectation of the agencies, how we have to develop the biological drugs, the biosimilar drugs against the biological drugs. So what type of data comparability required, we will have a complete picture. And we have to refer the respect to ICH guidelines as well. So in this way, we have to develop the product. So here also choice of the reference product. So the reference medicinal product must be medicinal product authorized by EMA and basis of the complete dossier in accordance with the so-and-so application number. So Indian guidelines also same thing, and EMA guidelines also same thing. So when you open US guidelines, also will be the same. So there is nothing. The expectation is the product which is approved in the respect to US FDA. So that product considered as a biological drug. Against that, we are going to develop a biosimilar drug. So that expectation is to be procured the reference product from US. So that accordingly we have to develop. So again here, I refer these guidelines. What their expectations? Manufacturing point of view process, expression system. This will be option to the sponsor organization. They may be developed in the microbial. We can use mammalian also if support, or they may use E. coli. We can use yeast. There is we can choose that is a completely organization, sponsor organization decision. Manufacturing process, it may be different from innovator versus ours, but the end product should not be differentiated. The end product composition, concentration, and other all biological activities respect to wild representation should not be changed. Route of administration should not be changed. So we can take different manufacturing process. When you're studying physicochemical properties, when you're testing functional activities and target binding, impurities point of view, you should be similar to the reference product. So that is what we have to be develop our biosimilars. Yeah, is it clear to everybody? What are the expectations from agencies?
Yes. Yes.
Yeah.
So I have a doubt. Like,
yeah,
how
please.
do you get the reference product? Like, we go to the market and purchase a reference product for different batches and compare with our innovator product.
Yeah, it is available. We have to raise as a PO. It will reach to the respect to purchase departments. They will buy and give to us.
Like we have to, okay, we can get, suppose if the product is marketed in the US, so but now we want to develop in India, means so we have to get from the US market like that.
Yes, that is the expectation.
Okay, thank you.
Yeah, so the slides is visible to everybody.
Yes.
Okay, fine. Yeah, this is what the 8 to 10 years what I am the timeline I'm giving. So just imagine today we are starting R&D activities. So it will take 2 to 3 years. The cell banks we can procure or we can construct. That is also manufacturer choice. Or upstream and downstream technology we can buy or we can develop our own technology also. And similarly, analytical methods also we can develop or we can adapt any readily available methods as well. But that should be validated in the respective stages. So conduct R&D studies, just imagine 3 to 4 years, and preclinical studies maybe 1 year, clinical studies 2 to 3 years, and getting permission from marketing authorization maybe 1 or 2 years for the review process, and launching of the product another 1 or 2 years. So like this, it may be 8 to 10 years of time period for the product start from development to reach into the commercial distribution. Yeah. So now why we had to discuss technical sessions prior to talking about marketing authorization or regulatory pathways. So coming out of 60 sessions, so we should talk about, we are talking especially for the regulatory CMC. We are talking for Monica. Yeah. So we are talking about especially quality module. So which is the data which is from R&D, which is from clinical, which is from commercial manufacturing should be placed in the dossier. The data like we can talk about process development reports or process development, process characterization, scale or model experiments, and method qualification reports, stability reports, stability data. So with respect to R&D batches, with respect to clinical batches, with respect to commercial batches, everything has to be parked here. So we will refer the M4Q guidelines, which is with respect to model 3. So that will be give us why I have to discuss technical sessions. To write the dossier, we should understand first technically to be strong. So there is a department like regulatory sciences, the people who write the dossiers. Similarly, in module 4 and module 5, there is a pharma people. So they having knowledge of pharmacology, so that there is a medical writers. In general, we'll call it a medical writers. So the hospitals will generate data, they will dump the raw data. By seeing the raw data, the medical writers will start the medical writing, especially on the preclinical reports and the clinical reports. That is how today we are getting job opportunities to work in the regulatory departments. So in the quality side as well as clinical side. Yeah, so those are in, especially we are talking about common technical documents. We will have 5 modules. So module 1 is a regional information, nothing but it is having only administrative informations, licenses, the applications, and the cover letters. It is like a overview of the what we have it containing. And module 2 is a like it is an index. Again is that module 3, module 4, module 5. So the reviewer, when you submit module 1 to module 5, a reviewer won't take up to review directly module 3, module 4, module 5. He will see or she will see module 2 what is happening. We are giving here quality overall summary. That means what is the batch scale, what is the batch definition, so what is the batch size and related to the quality, how many batches has been placed, the stability, and what type of study has been conducted. So everything is given a quality overall summary. When you mention there is a 5,000 liter scale of batches having 3 validation batches with respect to drug substance. So there is a section number so and so here. The reviewer will go to the directly validation data verification or review. When you mention there is a 6+6 studies of biosimilarity assessment in the respective section, the reviewer will go to directly open that 6+6 study report of biosimilarity. They will review it. Similarly, so when you mention the stability with respect to drug substance, with respect to drug product having 18 months or 20 months stability. Yes, Devashipla.
Sir, I have a question over here. Like the CTD will be part of the. At what stage will this come? Will it come in the NDA, the post the clinical trials?
Start to end will call as it is a CTD format, because what are the data we are going to submit in the US or regulated markets, we'll submit in the CTD formats only.
Okay, and so then when we are filing for the IND, that is right after the preclinical, right? So
Yes.
will that be in any particular format, or when we are
different we'll call CTD only, but it is having different filing application. So accordingly we have to draft it. We'll having minimal data. When you go for investigation new drug filing, we'll having R&D data and having preclinical reports only. When you're going for marketing authorization or new drug application, having all 5 modules. So when you're going for preclinical, we'll keep module 3 R&D data, module 4. And prior to preclinical studies, we'll have only R&D data. Then we'll go for preclinical studies. Once you want to work clinical, we'll keep these 2 data and we'll go for here. Once it is complete, we'll park all 3 modules. Against that, we'll write the overview, and against that, we'll place the regional information. Then we'll make a dossier into 1 to 5. So initial stage development here, it may be bits and pieces, and coming to this stage, will become all 5 modules in the structured way.
Okay. Understood.
Yeah. Got
Thanks.
it?
Yeah. Thanks.
Yeah. So this is how we have to develop. So we are going to focus more and more on the model 3 and quality overall summary and related to the filing pathways. So we will see at least 10 different countries filing, as well as completely model 3 different molecules like monoclonal antibodies, insulins, and vaccines. So these are the 3 industry leading molecules, including cell and gene therapy also is a separate session. And so going to discuss on this type of molecules. So we'll get the job opportunities and knowledge with respect to this type of biological molecules, so that we can go very clearly filing point of view. Yeah. So talking about CTD structure, what our previous slide I explained, this is the CTD structure. If the agency, this has been introduced by ICH regions, US and EMA and Japan. So commonly they made this type of format, and other rest of the world, they can follow similar path of those submissions. If the agency is asking to submit the CTD format, we should be submit in this way. And there are certain countries, they are asking to submit in the different their country format or their region format. So nothing but they're asking to change in their country specific. There is not going to change any technical content, only change with your any specific the terminologies or names or maybe numbering system. So that will be different with specific the country. If country is asking to submit in the country specific, then I have to modify, then we can submit it. So no need to generate any additional any technical data here. So like this, there is a called ACTD structure. So CTD, ACTD. So A stand for Asian countries. The Asian country, there are 10 countries. So those countries will be accepted in the ACTD format. So compared to CTD versus ACTD, A stand for Asian countries, associated Southeast nations. So there are 10 countries like Singapore, Malaysia, Indonesia, and Thailand. Like that, there are 10 countries in that region. So they will accept in this format. So this is older than this. So they have part 1, part 2, part 3, part 4. So one module is less compared to here. What is that less? So the module 2, which is providing separate overview, that is not provided here. So part 1 here, module 1 versus part 1 remains same. If you see module 1, registration, regional information. Here, part 1, administrative data, which is so related to the introduction part or maybe table of contents, application forms, labeling. Same content will be here in the especially module 1. That is what is called as a non-technical part, not a part of common technical documents. It will be vary country to country. The dossier which is made for the US submission. So you can module 2 to module 5, same thing will be there. Same dossier can file into the EMA by changing only module 1. So module 1 will keep with respect to US filing related activities. So when you want to same dossier want to submit to the EMA, so we'll place the data respect to module 1 change only. So module 2, module 3, module 5, there is nothing changes. Where you want to prepare the prepared CTD, want to convert it into ACTD. So what we have to do, part 2 is nothing but a module 3. Part 3 is nothing but module 4. Part 4 is nothing but module 5. So that the technical data is not going to change. Only the naming will change. So accordingly, we have to convert the dossier. So very easy. This is also one of the separate session for us. Once we understand the complete requirement of CTD, then we'll start conversion the ACTD. So this is how coming days we are going to learn the prepared CTD versus how we can convert into ACTD. So we can easily make it. Is it clear to everybody?
Yes.
Yes.
Yeah. Yeah.
Is
Yeah.
ACTD is applicable for India as well?
India not going to do it. Not going to take it because not part of any ACTD regions. India is a separate in the CTD format they're asking.
Okay.
Yeah. Yeah. So now we are discussing about molecules. So example, if you take out aspirin, is a chemical drug product, which is a small molecule. You can call it a small molecule. What are the number is given name versus number? This is the molecular mass. So one idea is the Dalton. So aspirin is a simple chemical drug product and is a easily to make it. We can make this product identical molecule. So already this is a branded product is already registered long back, so that we can make a generic product. So there is no requirement of any preclinicals. There is no requirement of any clinical studies when you want to conduct this type of manufacturing product. So we can perform bioequivalence studies and we can launch the product very easily. Within 2-3 years, we can make this product type of small molecules. When you come into the large molecules, which are the biological source molecules like insulin. So 5808, which is the Daltons in the molecular mass. You can imagine 180 versus 5808. How much differentiation? So it is having around 51 amino acids and erythropoietin around 30 some amino acids is there. When you come to the monoclonal antibody, around 30 some molecular weight KDA, around 30 KDA is there, 30,400 Daltons molecular weight is there. When you talk about monoclonal antibodies, around 150,000 Daltons are there. This is how the molecules complexity is difference from this versus this. So making this type of biological products very difficult for the manufacturers. So that is how this can be called as a biosimilar, not a identical molecule like chemical drugs. So how we can generate this type of molecules? So very easy. So which you are talking about pharmaceutical products like aspirin. So the size which is molecular weight less than 1000 Dalton can be called as a small molecule, and this will be mainly chemical synthesis. So product A will be go for conversions, any reactions, and any reagents will be used it to convert into product B, and that can be purified. Then we'll go for preparation of the final product in the form of any oral solids or any liquids or any injection form point of view. We can develop it, and it can be dispensed by usually retail pharmacies. Easily, if you go and ask, they can give directly to the patient or to the people. Easily can be get it. Where you're talking about biological molecules, large molecule, you can say greater than 10,000 of your Dalton of the product. Insulin, some cases we can call as a peptide also. The thumb rule is the molecule which is having 40 amino acids number will be falling under peptide category. Insulin is having around 51 amino acids, so we can consider as a peptide molecule also. Yeah, so this will become culture of living cells. So going to use microbes or cells. So by using this living cells, we can produce this kind of molecules by using a recombinant technology. And some vaccines, natural available microbes can be used. We can produce a secondary metabolite from directly non-recombinant source also. So often injected, either can be injected, can be infused. The treatment will be like you can take up monoclonal antibodies or insulins. We can inject intramuscular, or the erythropoietin or maybe monoclonal antibodies. It can be injected or can be infused to intravenous systems or any other route of administrations. So generally this can be prescribed by doctors or in the hospitals. So we go and ask directly in the hospitals, in the medical stores. They won't give it us unless without seeing doctor prescription should not be delivered. So like Herceptin, which is trastuzumab, so using for the chemotherapy, especially for the female bodies for the breast cancer. So this is how the molecules can be differentiation between small versus large. Yeah, is it clear to everybody?
Yes, sir.
Yeah.
Yes.
Yeah. So coming to the insulin, which you are taking a example of one of the mode of action, how it will be. So the insulin molecule. So can anyone add up any idea? So what is the insulin mechanism? Yeah, you can add up whatever you know regarding insulin. You can add up. It's like interaction should not be as a one side. Please add up. The people who are working the insulin can add up. Yes, Raja Reddy.
Like you're asking about insulin.
What is the purpose? What is the purpose? What is the mode of action?
The purpose is for control the sugar levels in the body.
Correct. Yeah. So if you're not given insulin injection to the patient body, what will happen?
Sugar levels may increase, or it won't be controlled in a state of.
So there will be sugar levels will be imbalance, right?
Yes.
Okay. So blood sugar it will be generally regulate the blood sugars. Yeah. So we'll take up into the session inside. Thank you, Raja Reddy, for your inputs. Yeah. So generally in the human body, what will happen? So when as a human, we take in any mixed meal, carbohydrates, proteins, so reach to the digestion system, so reach to the intestine. So what are the digested food going to convert in the glucose molecule? So the converted glucose, when you reach to the pancreas, which can be generate the insulin synthesis. So there is in pancreas, there is a beta isolate cells, which can be synthesized insulin. So insulin is like one of the stimulator, we can call it a enzyme kind of activity. So it will be activate to take this glucose molecules in our body tissue cells. If this insulin is not going to recognize to take up this glucose molecules, our body cannot be take up. So when body is not going to take up, this what are the generated glucose molecules will be soluble in the blood circulations. So automatically the glucose level will be increased. When glucose level is increased, there is a imbalance will be happen. So automatically lot of disorders can be happen. So that the insulin should be timely synthesized and timely the sufficient quantity should be available to carry out the balance of the regulation of the sugar levels. So this pancreas, that part of our beta isolate cells, any genetical disorders or any aging issue, sometimes may not be synthesized insulin. When the insulin is not synthesized partially or maybe fully, so the patient will be insufficient amount of insulin, so required to give artificially by injection. So now producing this insulin older days, there was a chemical methods was using. So those giving many side effects. That is the reason. So the side effects creating chemical synthesized products are replacing with biologically synthesized products. So how it will work? So this is the how the mode of mechanism. So this is the cell surface where the glucose level uptaking will happen. The insulin binds the receptor. So how the glucose insulin which is synthesized here will reach to the body tissues. The receptor will be recognized here. When it is recognized here, the glucose transportation will be open. So how it will happen? The blood sugar regulations after we eat, the blood sugars levels will be raised generally. So insulin is released to the help to lower these levels by moving the glucose into the cells. If energy storage also can happen. So when insulin production or function is impaired, as a diabetes or blood sugar levels can become dangerous to high, leading the various health problems. So it is required to control it. So how we can control? So we are going to make a biological medicine, which is our body, how we are synthesizing a biological product. Similarly, the insulin is going to make it exactly copy of this. So that will be prepared by using microbes. We'll make it the similar product of biological insulin, so that we go to the patient body. This is a sterile liquid solution of insulin molecule. So when you given certain dose to the certain route of administration, so that will be around 4 pH, which is in the acidic form. When you inject into our body in the intramuscular systems, due to our body fluids, neutral condition of pH, that will form as a precipitates. That precipitates will be slowly released into the body circulations. So it will reach to our body where the insulin, the glucose take up cells will be there. It will reach to that surface. Once it is reached to that surface, automatically glucose transportation will be take up. So all biological activities, energy generation will be happen as usual. So that is the reason when we identified there is a patient suffering with insulin diabetic condition. So it should be prior to meal or in a day required to give the sufficient quantity of insulin to the patient body. So when it was identified there is a insulin insufficient quantity or completely not generating in the particular patient body, so we have to inject them. So this is how one of the case study going to discuss in detail in the coming sessions. So how to develop this insulin molecules by using microbes. Accordingly, we'll discuss cell banks related one and upstream related one, downstream formulations, analytical methods. How to file these kind of products also we are going to discuss. So similarly, maps will take up another case study in coming sessions. So there also maps will be discussed, and this vaccines also will be discussed like this. Are we clear about this point of view? Why we are discussing about insulin?
Yes.
Yeah. So the insulins, there are analogs. There are 5 to 7 analogs in the market. So there are insulin glargine, which is the blockbuster molecule. So is currently approved in the US as well as a biological drug, as well as a biosimilar drug, as well as a interchangeable biosimilar drug. So taking into towards that. So what is the difference between biologics versus biosimilar? So we'll clarify in this slide. So biological drug is nothing but which is coming from living source. That is what the statement is giving. Biological products are regulated by Food and Drug Administrations, are used to diagnose, prevent, treat, cure the diseases and medical condition. Biological products are diverse category to products and are generally large, complex molecules. This is how we have been seen the differentiation between molecular mass, chemical drugs, and insulin products, and erythropoietin, as well as the respect to monoclonal antibody. Vaccines, if you compare, those are further more size of the molecules. So these products may be produced through biotechnology in living systems. That is how we are going to learn how to produce this type of molecules. Such microorganisms like plant cells or any animal cells are any often more difficult to characterize than the small molecule drugs. So there are many types of biological products approved in the US, like United States, including therapeutic proteins such as filgrastim, monoclonal antibodies such as adalimumab, and vaccines such as those are the influenza and tetanus. There are many kind of products. I'll show it how many products so far approved in the US market. So what is the reference product? The reference product is nothing but which is first time is registered in the market. So nothing but the application path which has been used 351 application A. So which is registering for the new biologic. So reference product is a nothing but new biologic. A reference product is the single biological product already approved by FDA against which is proposed to the biosimilar, which we are trying to make the biosimilar against this product is called as a reference product. Yeah, sorry. Yeah, is it clear to everybody what is meant by reference product?
See,
Can you
maybe
please come
sometimes
again on that?
Yeah, so reference product is nothing but so far it is not there. It is the first time registered in the US FDA. Got it?
see,
Okay.
maybe like why we have to consider only USFDA? Maybe I will.
Not only US FDA, I'm talking about when you're taking application pathway as a category of 351. That's part of you, I'm telling. Okay. It is can be approved in the EMA as well prior to the FDA. It may be approved in the other regulated markets or any other countries. But the preference will be given the product which is approved in the FDA or EMA, that will be more considered for the development of biological drugs against any biosimilar drugs. Clear?
Okay, like see, sometimes maybe in Indian market, for example, the new drug might be generated and the clinical trials, everything has been done, and the drug may be the efficacy, effectiveness check, it is working for curing the some particular diseases. Can't we consider it as a reference product?
So if you're looking for Indian market, then we can take it into that point of view. When you're talking about US FDA, definitely we should consider reference product which is first time registered in the US. That should be considered as a reference product market wise. So which is registered in the India, we cannot take it as a reference product to file the product in the US. Got it, Raja?
Okay, okay.
Yeah. Yeah.
So it can be said as an innovator drug, right?
Nothing but innovator. Either you can call it a reference product or innovator drug or a RMP. These are all will be as a same terminology. The terminology will remain same.
Okay.
Yeah, fine.
Follow-up question. Hello. Follow-up
Yeah, please,
question on
please.
that. Any reference product do India have which is approved in FDA?
No, as of now, no. Okay. Yeah. Not there. Okay. Yeah. So we are all working in the Indian market, biosimilars point of view. Yeah. So now biosimilar product. Yeah. So biosimilar product, which is developed against this reference product, which is called as a biosimilar product. The definition says that biosimilar is a biological product and that is highly similar and has no clinically meaningful differences from existing FDA approved reference product. So which is already approved this product against that. So quality comparison when you did, it is a similar. When preclinical and clinical, when you perform the studies, also similar. There is no meaningful differences. There was no any significant changes. So that product can be called as a biosimilar product. So that is what we had to take this as a reference product. Against that, develop the similarity assessment, analytical point of view, and clinical comparability, preclinical comparability. So make a complete package of biosimilar. So when you go into file in the US with complete package of biosimilar application, so easily can get the approvals when quality and preclinical, clinical, safety and efficacy data is comparable. Yeah, so that is what we call as a biosimilar product. And taking into another one, which is called as a interchangeable product. So do you have any idea about what is going to be interchangeable biosimilar?
So we can use the product instead of the reference product.
Exactly.
Patient.
Yeah. So any manufacturers are doing right now interchangeable biosimilars in India or any other way?
Biocon.
Yeah.
Insulin glargine.
Yeah. Exactly. Yeah. Great. So the insulin glargine is the claimed as a interchangeable biosimilar in the brand name of Semglee. it is a partner product of earlier Mylan versus Biocon has been made and got the approval as a interchangeable biosimilar. So what is the benefit here? The interchangeable biosimilar means, so the innovator product, so Lantus is the brand name of Lantus, launched in the US and it was approved as a reference product, innovator product or RMP. Again is that has been developed biosimilar product and also got registered. And if doctor refers to use reference product, the patient or the pharmacist should be use this product only. The doctor is preferred to use biosimilar product, can be used biosimilar product. Let's imagine the doctor's prescription is given to the patient to buy the product, so and so product in the medical store. The patient is approached to buy this product, but stock was not available. So either he has to be wait for the stock or we have to go for any other medical store. So the US FDA body has been developed the application pathway along with the biosimilar pathway. In the same application, we can add up additional clinical studies and can be make a interchangeable biosimilars. This interchangeable biosimilar, when it is in the market, approved market, so when this product is not there, the pharmacist without approaching to the doctor, he can dispense this one. And in front of patient, he can place reference product versus interchangeable product. Either way, we can choose it. So what is the difference here? What is the benefit going to happen to the patient? Just imagine the price wise. So this is $100 in the US market. Just imagine it may be more or maybe less, $100. So going to biosimilar, it will be around 20 to 25%. Just imagine $25. So the difference between these two almost 4 times is high. So the US especially preferred to biosimilars. The reason is the government bodies going to take care of their public health, so that they are going to invest huge money on the healthcare. So one patient where they're investing money for $100, they can invest the $100 for the 4 patients. They can save the 4 patient lives. There is no any quality comparison here. But just imagine last 10 years, the patient has been adapted to this medicine. So the patient can take up or may not be take up, there will be doubt. For that reason, the sponsor organizations can do additional clinical studies to file as a interchangeable biosimilar. There is a switching studies that will happen, especially for the interchangeable biosimilars, that will be conducted especially in the clinical studies. So the interchangeable biosimilar, once you want to claim it, so the US FDA, especially in the United States, biosimilar seeking interchangeable status must be meet additional clinical trial requirements. Beyond those needed for regular submission biosimilar approvals, the FDA evaluates interchangeable under 351(k) application pathway. For biosimilar also same thing, and respect to interchangeable also same application. So we are going to add up. So initially the organization will be trying to add up as a biosimilar, then going to add up the additional data switching studies to the existing product. So public health as per the PHC Act, Biological Price Competition and Innovator Act. So this is the biosimilars they are preferring to low cost, and sponsor organization can be develop additional data, can file as a interchangeable biosimilar. The cost will be increased. How it will be increased? I will take a bit to another slide before going to that. Yeah. So here, what is an interchangeable biosimilar means? Interchangeable biosimilar is a biosimilar to the reference product. First thing, it is nothing but a biosimilar and expect to produce the same clinical results in the any given patient. This is how required to conduct in the during switching studies in the clinical. So can be submitted at pharmacy level without prescribing. Approvals are subject to the state of the laws. So this is how government body has been produced the rules and regulations for the interchangeable biosimilars. The pharmacist can be deliver the product, or the patient also can choose either interchangeable biosimilar or it may be reference product for usage. And switching studies, what we are going to conduct in the clinical means study design and features multiple studies, at least 3 switches between reference and biosimilar. Compare switching group, reference to biosimilar, and non-switching group, reference only. So this is how we will develop the data on the clinical. So quality wise, no changes. Whatever the biosimilar has been developed, the quality data remains same for the interchangeable biosimilar also. And preclinical, no changes. Only changes with the additional data going to generate. So focus on the PK/PD endpoints, immunogenicity point of view, safety point of view. So this is how we'll generate additional data to file the innovator product to increase the preferable product usage in the market. So here FDA expectation switching must not result great risk in terms of the safety, any reduce the efficacy or increase immunogenicity. This should not be happen. And special consideration, sensitive population may be required. Example, any compromised, any pediatric conditions or any chronic conditions, insulin, adalimumab are often preferred for interchangeable studies. As of now in the US market, these are the two products approved as a interchangeable biosimilars. One is insulin glargine, another one is a adalimumab. And here submission requirements for interchangeable will apply 351K application for the biological license application. Analytical similarity package, what we deliver for these biosimilar products, same package will be applicable for the interchangeable biosimilar. Clinical study will have additional data, which is having a switching study reports. So labeling proposal different compared to biosimilar because
PKPD study.
Yes, that is a part of our package completely PKPD studies.
I
And-
have a question.
Yeah, please.
What is RLD then?
Sorry, RLD.
Hmm.
Reference license drug.
Yes.
So
Then how
we
does
use-
it fit into the last slide? Mani, can you just go to the last slide and just let us understand?
May I know what this?
Ananya?
Oh, okay. Yeah, please tell me.
RLD and relation with this.
Interchangeable.
Meaning what is RLD and does it have any relation with these three listed products?
It means reference license drug or reference product or reference medicinal product or innovator product. These all same terminologies, same. There is nothing differences. Either you can call as a RMP, RLD, innovator product, biological drug.
Okay.
Yeah. So submission point of view, these are the data required for the submissions. Let's imagine we submitted the product and got the approval. So what is the benefit to the sponsor organization? The sponsor organization, just imagine Lantus, which is the company has been made, Sanofi, and insulin glargine. The price was a reference product around $292 when it is approved in the US. And the Semigly, which is the brand, has been approved in the biosimilar pathway, and we got it insulin glargine around $99. So almost all 30% only cost of this product. So 35% only against this one. So where the government body can be buy at $292, now this Semigly brand of insulin glargine can be buy at $99. So it can be served for the 3 patients instead of 1 patient. So this point of view, government body will be looking to go for this kind of products so that they can save the many lives. Now, when we have been conducted additional switching studies and as want to file as a interchangeable product, there is a Semigly, same brand and having a insulin glargine. So when it was submitted additional switching studies data and FDA government has been approved in the 2021, which is costing around $269. So almost all 292 versus 269, around 90-80% is cost of the equal to that. So because of the product has been proven, absence of this, it has been given similar efficacy, and it is switching studies has been proven it, so that the price was increased. So the Biocon Biologics has been made initially a Semigly brand, and they got approval for the biosimilar pathways. In the same pathway, they uploaded the data of respect to clinical studies and got approval as a interchangeable biosimilar. So when it is market is labeling with this interchangeable biosimilar, the patient, the pharmacist can be dispensed this, This is choice of without any doctor prescription. We can take either this one or can be this one. Options of this one we can take up, or presence of this one also we can pick up this one. So this is how we can get a approval from US FDA for the biosimilars, biologics, and respect to interchangeable biosimilars. The cost-wise differentiation. Is it clear to everybody?
Okay, if
Yes.
both are not there, reference drug and interchangeable drug, then can patient go for a biosimilar?
Yes. Doctor has to be prefer it. When you want to take a biosimilar, doctor has to be prefer it. It should be written in our prescription. Then only the pharmacist can be deliver it.
No,
If pharmacist want to dispense the biosimilar drug, Doctor has to be written in the document, the prescription.
Okay, doctor might have reference like no biologics, like no maybe insulin glargine, for example, innovator drug, and then when patient come out, like when he search for in the medical stores, those both are not available, for example.
Then whether wait for it and go back to the doctor, and doctor is given preference to organize this biosimilar, then only the pharmacist can be dispensed.
Okay.
Yeah. Fine. Okay. Yeah. So now.
Yeah, yeah. In this slide, last two rows are same only. No? I am not getting the difference.
See. So insulin glargine, we're talking about interchangeable biosimilar and authorized generic launched. It is a generic launched. This is a biosimilar launched.
Okay. Okay, so price again came to $99.
Yes. Generic version versus biological versions. Yeah. So when you're talking about the molecules, which are talking about insulin, which are talking about any maps or any vaccines, recombinant vaccines, how we are going to generate it. So the data, how we can generate with respect to development phase. So that any microorganism cannot be developed what we require. So what we have to do by using biotechnological methods. So we have to go with recombinant technology, cloning methodology. So we have to isolate genome, which is containing gene of interest. Just imagine we want to produce the insulin molecule. So when you want to insulin molecule want to develop or grows, so we have to identify the genome. From that gene has to be target to isolate it, cut this gene, go with recombinant technology and vector construction, and go to the selection of the host cell. The host cell can be the sponsor organization choice. Someone can be use E. coli, someone can use CHO cell, someone can be yeast. There is a completely manufactured choice. No one regulatory body will ask for the which type of they will ask to which type of expression system has been used, but required to use only similar type of expression system. No one will be demanded. This is a complete organizations do different, and they can choose their point of view. Once we construct the expression system, we will go for cell banking. So the cell banking like master research cell bank, then go for master cell bank, then go for working cell bank. So why we have to go with this RCB, master and working cell bank, that we'll discuss tomorrow, tutoring system. So once we made the cell banks, we'll go for R&D development. We will see the productivity of the product, how we are going to develop that in R&D stages, and how will be produced the small scale, and we'll see the repeatability, quality and quantity wise. So we'll make a purified bulk drug and go for the formulations. Once we made this product, we'll go for animal studies and we'll go understand the preclinical point of view, clinical point of view, then we'll go for large scale commercialization. So now this reference biologics, using reference biosimilar and interchangeable products, how we can take up into patients and healthcare providers can be concentrated on safety, effectiveness. When you want to develop either is a biosimilar or interchangeable, so we have to see safety, effectiveness and respect to quality data. So that is how initially I explained biosimilarity can be proved, safety, effectiveness and safety. So biosimilars and interchangeable biosimilars, so product and prescribe the by name just for the reference product. So here the biosimilars and interchangeable biosimilar products can be used as a patient who have previously been treated with the reference product. So we can use interchangeable or biosimilars. The patient has been already treated with the reference product. So treatment, any experienced and any patients who have not been previously treated with the reference product. That is treatment native. So here interchangeable biosimilar may be substituted for the reference product without intervention of the prescribing the healthcare provider and subject to state law. So when the medical doctor has been so reference product has been preferred, so it will reach to the medical store and pharmacist can be dispensed with respect to reference product. If it is not available, without prescribing the doctor prescription, he can dispense interchangeable biosimilar. That is the benefit of availability of the productivity of the interchangeable biosimilar. So that is the reason FDA also approving. The reason is drug demand should not be happening to the patients. So drug shortage should not be happen. At the same time,
one question here. Why
yeah,
the leniency? Why this leniency for interchangeable biosimilar? Why this.
Why they required it?
Yeah. Why it is like, without prescription, like why this? Because mode of action, everything will be same, right? Why
Everything
this,
will be same.
Yeah.
Everything will be same. The additional data will be available, which can be replaced the innovator product usage.
Yeah. Why it is special provision
Requirement.
provided for interchangeable? Yeah.
The reason is to become alternator to the market. If suddenly the existing molecule is stopped or suddenly you raise the price, so it is a burden for the people, burden for the government bodies, so that if having an alternate competitor, so they can prefer the low cost one. For example, Biocon is selling at $270 and Lantus is selling at $299. Just imagine Lantus tomorrow increase the price to the 350. So definitely they will buy the biosimilar product of interchangeable biosimilar product of Biocon one. Or just imagine e-stop. They should not be happen due to the e-stop because of any XYZ reasons. So we'll have a alternate product in the market so that easily available to the patients. So when only one product, it should not be monopoly. You should be having a multiple choice of the selection of the productivity in the market.
But this is only in the point of economy. What about other, safety, efficacy, and all like?
That's what already proven. No, that is what safety, efficacy, and respect to effectiveness already has been proven in the respect to clinical studies, preclinical studies, and quality data already proven. Then only that product has been approved.
But biosimilars, regular biosimilars will not be dispensed without prescription.
Yes.
But in this case, it is without prescription.
Choice.
It is why it is?
No, not without prescription, without doctor intervention. He will be written already reference product, but the stock is not available. Then the pharmacist can be dispensed either reference product or can be interchangeable product. It is an option, and it is two products available in the pharmacy, and patient also can choose whether you want to buy Lantus, whether you want to buy Biocon product. Such a way we can option will be there. But in case of biosimilars, if doctor is written in the document, the prescription, we have to use same thing. Clear?
What about the issues? In conflicts or any issues arrive after consuming these interchangeable.
That's
anything goes wrong in the wound.
That's what clinically already no meaningful. The statement was on clinically no meaningful. That has to be proven before approval, not after approval. Even though after approval, we'll go for phase 4 data collection. If any such kind of any incidents or any adverse events has been raised, we should be taken action immediately.
But
Right?
again, I see only economical point of view explained, but like what is the biosimilars and
before product approval, before product get approval in the market itself, we should ensure safety, effectiveness, and as well as quality. Then only product going to approve it. Once it is approved as a market point of view, economic point of view, but we should not compromise with our quality. That should be proven. Then only the product can go to the market filing. Otherwise, it will be stopped. Without product quality, we should not consider it as either biosimilar, it may be interchangeable biosimilar. It has to be proved first. After ensuring
same with the biosimilar, right? Biosimilar also will go through the same type of scrutiny.
Same pathway, but don't have any switching studies.
Okay.
Yeah. So these are the listed products, which B stand for biosimilar, I stand for interchangeable biosimilar. So we're having different products in the market. Like
I have a doubt here. So the biosimilar product which undergoes this which undergoes this switching means we call as a what is called that interchangeable biosimilar. Is that way?
Biosimilar? All interchangeable biosimilars are biosimilars, but all biosimilars are not interchangeable biosimilars.
Yeah, okay. So biosimilars which undergoes this switching study means it comes under interchangeable biosimilars.
Exactly.
Yeah, okay. Thank you. Got it.
Yeah. So there are different category of the products, classes. So we can talk about supportive care medicines, oncology products, and autoimmune products, and ophthalmology products. So if you see, there are 41 biosimilars, and so there are marketing around 29 products, and in that there is a insulin glargine have holding interchangeable biosimilars by 2 organizations, and adalimumab having one of the organizations as a holding as a interchangeable biosimilar, and ranibizumab also having as a interchangeable biosimilar. So this is an overview of biosimilars, and you see very 3 products only in the market, and among the 3, only 4 organizations having a interchangeable approval products in the US authority. Yeah. So now why seek interchangeability means, so should talk about interchangeable biosimilar products can be substituted for the reference product at pharmacy without intervention of the prescribing healthcare provider, nothing but doctors. So subject to state pharmacy laws. So here pharmacy level substitution, how it will happen. So the doctor prescription, the reference product, he written the prescription as a so-and-so Lantus to be used for the patient who is suffering with the diabetic condition. The script goes to the pharmacy and the pharmacist substitutes interchangeable biosimilar for the reference product. So the reason 1, patient is looking for the low cost, and the reason 2, the product is stock is not available, so that he can dispense another interchangeable biosimilar product. The patient receives the interchangeable biosimilar without any doubtful. So this is how benefit for the product has been registered in the interchangeable biosimilar. Yeah. So now taking into quality data. So what we developing for the biosimilars? So analytical, highly similar, interchangeable also same data. High product quality standards, same thing applicable for the interchangeable. No clinical meaningful differences for the biosimilars, same thing applicable for the interchangeable. But taking into interchangeable separate zone, it should be additional studies to be given at any given patients, a risk of the alternating to the switching studies to be conducted. So this data required to generate access towards the biosimilars, so that it can be become as a interchangeable that can be go for filing in the FDA. We can get a interchangeable approvals. So here applicants requires licenses as a biosimilar or interchangeable biosimilar. So the application will be 351K, so that there is an option to select biosimilar, interchangeable. So when you select interchangeable, required to submit this all clinical data which is generated. When you're talking about biosimilar, limit with this. When you're talking about interchangeable biosimilar, complete of this. And an applicant may be first approved as a biosimilar, but later seeks the interchangeability. So this is how our Biocon Biologics has been done, get the approval in the market. And the analytical similarity and product quality standards, the same biosimilars, interchangeable biosimilars, there is no any differentiation. And the statutory criteria for related to the potential and any substitution without the intervention of the prescriber. So this is how we'll get a benefit from the approval authority. So we can use reference product, can use interchangeable biosimilar. So like the way we can get the approvals and usage of the medicinal products in the US. Yeah. So to develop this product, so we want to develop these products. So we have to use reference guideline ICH M4Q to develop the dossiers point of view. When you're looking for to develop the dossier, we should know what is the requirement. It's not like that today in 2026 we are developing R&D, and 2032 we are trying to file the dossier in the US. By that time, if you see there is a data is insufficient, then it is a difficult for the filing. So the regulatory strategy, the people who are working in the regulatory department, I shown the role and responsibility in day 4 demo session. So as a regulatory CMC strategist should be prepared according to this requirement. So as a part of filing requirement. So as of now, the ICH M4Q R1 is going to revise. So required to generate additional data, like there is a many things going to discuss in the during our dossier preparation. And whenever there is a technical session is there, we are going to refer M4Q, and M4Q R2 is a under draft version. So going to revise. So once it is approved, every organization should be change their dossiers in this format. There is a lot of corrections going to happen. And the guidelines for the Module 3 CMC regulatory strategies. So here we'll refer this guideline. We'll understand what is the requirement, why you have to discuss about particular CMC sections. And the second guideline, which we'll talk about safety guidelines. For the as of now running guidelines, same thing. We'll go for PCT regulatory strategies and data generation for the filing activities. For the biologics versus biosimilars, anything you want. And refer to the M4U guidelines for Module 5 for the clinical trials, regulatory strategies, and data generation for the filing activities. So these two guidelines is a reference and for the overview. But our sessions, around 40 sessions, will talk about this complete CMC data, and 10 sessions will talk about how to write the dossiers, and 10 sessions will talk about completely filing activities in the different countries. And there is another guideline part of our multidisciplinary guidelines. M stand for multidisciplinary, and 4 is the sequence number. Q stand for quality, S stand for safety, E stand for efficacy. So there is a CTD organization. So how to organize these dossiers in the folder organizations? So when you want to submit this CTD dossier in the electronic format or maybe in the hard copies format, we should be create a folder structures. Is a complete leaflet or completely folder structures. So in this folder structures, we have to create the dossier. So how to dossier can be created in the either CTD format or ECD format. We have to refer these guidelines. When we are going to write up the dossier, we will refer these guidelines. Since we are going to talk about technical sessions, we are going to refer these guidelines. Yeah, is it clear to everybody?
Yes.
Yeah.
Yes.
So I will show the guideline. So how the expectation as per current guidelines. So which is the current guidelines is M4Q guidelines, which is produced on the upload on the 2002, which is currently running. And now this guideline is the going to change revision or is going to happen. So here the common technical documents, sorry. Yeah, so the common technical documents, so which is required for the filing activities in the ICH regions and which is prepared for the ICH point of view. If the organization or that particular country, organization means the regulatory body, can accept ICH format, we can submit the same thing. So every manufacturer, first they will be looking to the CTD format with respect to ICH requirement. So when you inline with ICH requirement, it easy to go for any country filing. So even the India also will look into the as per ICH, so that it will be fit into the India requirement. So the registration of the pharmaceutical products and for human use quality, there is a M4Q multidisciplinary guidelines quality module going to revise under R2. So this is last month we has been released on May 14. Now exactly 1 month is over, currently under public consultation. So people can be review and they can give their comments. The government body can be, if applicable, they can revise or can be same guideline going to publish it. So there is additional data, numbers going to change it, but technically there is few data can be add up, which is already part of development, or if it is not part of development, required to generate. But mostly the data will be available. We can modify that structures names and the numbering system. We can file the same thing in the respect to CTD format. If you see our current guideline, so we'll talk about with current guideline. So it is having model 2, model 3. So model 2 is a quality overall summary, which is required to see here. So which is the quality overall summary, and the quality overall summary is a summary for the complete model 3. So model 3, whatever we provide the data, that will be having a summaries, very minimal information. And here should be some 80 pages or maybe some 40 pages should be prepared. So accordingly should be given a the overall summary. So when you entering into model 3, so that is the quality which is required to talk about R&D to till commercial. So here having the model 3 having 4 parts. So when I open into 3.1, TOC generally called as a table of content, and 3.2 is a body of data. This body of data having 4 parts. In that, one part is a drug substance. 3 stand for model 3, 2 stand for body of data, and S stand for drug substance. So complete drug substance information to be draft the dossier. So to write this dossier, we should require a source documents. What are the source documents? When you want to write up nomenclature, or when you want to write a structure of the molecule, when you want to talk about general properties. So what should be required? We require the product characterization report. The product characterization report, which is having a the generic name or any compoundal name or chemical name or company laboratory code, and company laboratory code having company to company different codes, and chemical abstract service numbers which is available for the specific to the product wise. And during those development, how to get this number we will explain. And structure, when you're talking about structure, biotechnology products. So what is the expectation here? Schematic amino acid sequence. When you discussed about insulin having 51 amino acids, insulin human. When you're talking about insulin glycogen having additional 2 more amino acids, 53 amino acids. So we should talk about that amino acid sequence. and indicating glycosylation part. If applicable for that particular part glycosylation, required to write it. If not applicable, no need to talk about it. And disulfide bridges, disulfide bonds between the chain A versus chain B. So required to explain. Similarly, monoclonal antibody or respect to any vaccines. So any glycosylation site or any the disulfide bridges or disulfide bonds should be explained at what site. So what amino acid to another chain, what amino acid is corrected. It is inter or it is a inter. Other post-translational modifications and related molecular mass should be provided. So this is the expectation from regulatory body to write this structure related information with respect to product. So just imagine you are trying to apply the product as a insulin glargine, should be written a completely insulin glargine data, which we having a characterization report with us. Got this point? Yeah, please respond.
Yes.
Yes.
Yeah. So that is
Yes,
how we
yes.
had to know completely characterization to write the dosage section. And similarly, manufacturing. So when you talk about manufacturing, there are main section is a S2.2, 3 point module 3, and 2 stand for body of data, and S stand for drug substance manufacturing. Under that manufacturing, there are subsections like manufacturing address. So which we having a manufacturing license, which is brought by regulatory department for the manufacturing license. That license address, everything should be placed here. When you place the address completely, it will be reached to the regulatory body. They will note down this address. When they are coming for the audit or visit, they will come to directly this unit address. So when it is mentioned, so-and-so unit, so-and-so block, so-and-so room is going to happen drug substance manufacturing. So they will strike forward. They will come to drug substance manufacturing with respect to cell banks area, with respect to upstream, respect to downstream. And in case of formulations, in case of QC, everything should be available here. Anyhow, formulation will be separate section, but respect to drug substance analysis QC also part of it. So the address required to mention it as per the manufacturing license. Now talking about discussion of the manufacturing process and controls. So completely QBD has been how we have been developed the robust process. So QBD elements has been used. QTPP has been developed. Against that, CQ has been established. Risk assessment has been conducted. And CPP has been established. CM has been established. Control studies has been prepared. And design space has been created. So that all the QBD elements has been used for the process development. And we have been made a robust process which can be transferred the technology. Start from cell banks to reaching to the typically start with a vial of cell bank and includes culture, harvest purification, and modification reactions. Modification means any enzymatic conversions, and filling. The drug substance is going to fill into the any bags, any glass bottles, and storing at what condition, and shipping condition. So why we should talk about shipping condition means the drug substance after making into drug substance area, how to shipping to warehouse, or it may be drug substance is making in one unit and drug product is making another unit, another location, another country. So should have a shipping validation. So this is the expectation here. And similarly, completely the upstream process should be known, downstream process should be known, and filling process should be known with respect to drug substance. They are linked with other quality sections that should be known, and respect to guidelines, ICH-Q5A, which is viral clearance, cell substrates, and biological specifications. These all should be used while you're talking about your development and manufacturing. Then control of materials, cell banks and raw materials, input materials, all will be covered here. We should talk about completely cell banks and characterization. Control of critical steps. What type of controls you having? Intermediate, in-process analysis. Then process validation. That is how we have a separate sessions for the process validation to understand complete package, including hold times, including cleaning validation, including filter validation, and transport validation. Everything should be placed here. So should be required from data manufacturing side. And manufacturing process development, completely R&D. So that day we initiated the development activity at R&D. So as we regulatory persons, we should request to R&D provide the development reports. How we have been started the development history. They may started with screening, resin screening, or 1 liter scale, then 5 liter scale, then 10 liter scale. They come with the different scale approach. So how they made the manufacturing scale ups? R&D made done 20 liters. Then manufacturing made done 2000 liters. Then commercial further scale up has been done 20,000 liters. That entire life cycle management, we have to keep on update this manufacturing development. Then we should talk about characterization, protein characterization, structural elucidation. For the biological products, we should talk about product and product related substances, product related impurities, and process related impurities. How have we separated? And secondary, primary, secondary, higher order structure should be discussed. Post translation PTM should be discussed. Glycoforms, biological activity, purity, immunochemical properties should be discussed. So we should all aware respect to this mechanism when you want to write up a dossier. That is how the people will hire from here different domains. The person who having analytical background, they will hire for the writing of this type of sections. The person having manufacturing background, they will hire to write these sections so that they can easily develop the dossiers. Then talking about impurity characterization, also part of it. Then control of drug substance specifications should have a in-process specifications, lot release specifications, as well as stability specification. In-process specification will be applicable to place here, and respect to lot release, respect to stability will be placed here. Then analytical procedures, nothing but STPs. It may be product STP, it may be general STP. With respect to any other STPs, they are applicable to the test this analysis. We have to park it here. And analytical validation. That's what we conducted one of the demo session. And going forward, we'll talk about development. We talk about specifications. We'll talk about the JOS. We talk about batch analysis. Completely analytical package will be there. Separate CMC sections. You can see different guidelines when you're talking about characterization Q6B. When you're talking about impurities with respect to drug substance and elemental impurities with respect to solvent impurities, and Q5C biotechnological products stability, and Q6B specifications. Everything is referred. So this how will be refer respect to guidelines during this technical sessions, so that will give up to the requirement for the writing up to US level of submission. Then reference standards. There are types of reference standards, internal reference standards, or we can call it a primary reference standards or secondary reference standard. There are also two tiering system is there. Then container closure system, which we are going to use for the storing of our drug substance in the respect to containers. It may be glass containers, it may be PETG containers, it may be any single-use bags, it may be LDBA drums, or maybe any other type of containers we are going to use it. How it is compatible, store that, which temperature has to be store it, how we can ship it. So this and all will be required to place it here. Then coming to stability, the drug substance stability. Should we understand the stability point of view, where that product is stable at -20, -40, or 228. So which is the formal stability, and categorizing the accelerated condition, long-term condition, and stress stability studies, thermal, agitation, mixing, water stability. So these and all should be studied, and summaries to be given, and post-report stability commitment to be given, and stability data with statistical analysis, graphical or narrative write-ups, and tabular forms to be provided the data. So when you submit the agency, they will review first, they will understand the summary, they will see the comparison. So 0 month, 3rd month, 6th month, 9th month, 12th month. First year should be in the form of quarterly basis. Second year will be 18 months, 24 months, half-yearly basis. 3rd year onwards, the agency expectation is yearly basis. That means 36 months. As we are sponsor organizations, to generate extra data, we can add additional time points also during our initial phase. So according to Q1 stability guidelines, Q1B stability guidelines, this is drug substance stability, Q1B water stability, Q2A, and respect to the validation which you are going to use for the validations, and Q2B and Q5C. These are all required to refer during stability data generation. And similarly, drug product, whatever we see in the sections, similarly, product description and pharmaceutical development, R&D development of our biological drugs, our R&D development, then followed by commercial manufacturing, and followed by excipients. What we use the excipients, what are the controls? We having specifications, we have a procedures, we have a validation of applicable, and JOS area excipients using from human or animal origin required to provide a information. If no, we can say no. In case of any novel excipients that required to provide, then drug product completely, release specifications, stability specification, STP, validations, batch analysis, characterization of impurities. So here required to provide any nitrosamine impurities also, and JOS reference standards, container closure, and followed by stability. So this is how 2 parts. And 3rd part we'll talk about appendices. facilities, equipments, qualifications, and our advanced viruses, safety evaluation, viral removal, how we are doing, viral clearance, and followed by our excipients. And the fourth part is a regional information. Here, batch records, method validations, comparability. This is the biosimilarity going to provide here. And process validation, we can give the reference. If medical devices, if applicable, we'll give it. And during development, our R&D has been used. Any references, any literatures, that also can be provided. So this is how the expectation from filing activities in the as per ICH regions and other countries. So we have to be technically to be generate the data. That is the reason we have been developed in this structured organization so that we can go systematically. Chapter 1, we'll talk about raw materials and cell banks, complete package. Then upstream, here 3 products will be taken as a case study. Everywhere upstream related 3 products, downstream related same products of upstream development, and upstream, downstream, the same products, how to verify it and how you use the QBD. And we'll talk about stability required for the DSTP, equipment qualification required, cleaning validation required, and followed by process validation required, GMP, clean room area classification, ALCOA, audit point of view required, compliance point of view required, complete QMS package. Then regarding drug product development, 3 products, and we are using software also, JMP, another kind of also we are using, and analytical package, completely development, method transfer, method validations, reference standards, analytical characterization, biosimilarity, bioassays, immunoassays development, optimization. Then there is a cell and gene therapy, one overview session also there, medical devices. It is a separate sessions, but we are conducting as a just overview, and how to write the dossier, and followed by submissions. So CTD, ECTD, ACD conversions, how we can do it. Then Asian countries like Malaysia, Singapore is a case studies, and PCT application with respect to RCGM and IBSC board, how to file it, and CT application, CDSO, the reference guidelines. So when you talk about clinical trials, IMPD with respect to EMA, and pre-IND respect to US filing point of view, will take up. Model 1 case study, Saudi FDA filing, and domestic market, Indian market, complete package, IBKP, SUGAM, IBSC, RCGM, and related to the CDSO and GSE, Drug and Cosmetic Act will cover completely. And CTD dossier filing into EMA. Initially I mentioned centralized, there are different pathways. US FDA, then TGA, Australia, LATAM region, Japan, China, and MHRA, and MOH queries, how to address it. That is also one of the session. And variation filing will go for different regions, different countries. Then computer system validation. So this is how maybe it is reviewed by everybody. Is there any doubts to anyone? So that's all from my end today's session. We'll go for any discussion.
Whatever you have mentioned, that softwares for the CTD, will you able to give the demo?
We don't have any live softwares, Raja. So it is not possible to buy, or it is not at all possible. I will give the mockups. So I'll show how the software exactly screenshot. I'll show it. So it is like a one app kind of basis. So we can log in. We can go for buy. It's not possible to buy by anyone because those are the costly and not at all possible to get it by license by us.
Okay, thank you. And one more thing, when we are discussing about the validation batches, we one batch is the enough, you said, right? Like
no, not validation point of view, clinical batches point of view.
Okay, for the clinical, one batch is enough, you mean to say?
Also we can do, 10 also can do. That sufficient quantity is enough for one batch. We can able to justify. We can justify it. Not necessary to perform many batches.
No, like minimum 3 validation batches is required, sir.
If you are going to perform validation, then only required to generate 3 batches concept. When there is no validation point of view, we can enough to 1 batch also, sufficient quantity we can generate and go for clinical studies. Clear?
No, I didn't get
No.
it.
See, very easy. See, when you want to clinical trial batches, right? Are you able to see the screen?
Yes.
So our organization is not going to do any validation. Just we have to generate the clinical batches and go for the clinical studies. Okay. We can take 1 batch that can go for converted to drug product and go for studies.
Okay.
When we decided our process, everything is streamlined, everything is done, and want to having a biosimilar concept here. Okay. So we can take 3 batches. We can do validation. When there is no validation, there is no preferable number of batches to be taken here.
Okay, then that is the case. Like we will be taking a more time for going to the market, right?
Yes, that is what. So the organization should have a clear strategy. Instead of complete clinical trials without any validations, then get the approval from market, then we'll do validations, then wait for the stability and go for less stability data, launch the product. Instead of that, you plan for clinical batches, equal size to market, and go for stability studies and complete the stability. By the time marketing approval, we may complete 3 years stability. The moment we're getting launching product, it will go with 3 years stability, so that there will be no any variation filing. There is no any issue with less shelf life data.
Okay, when we are going for the first filing, how many 6 months is enough, or like 12, 24 months?
We can submit, say, we can submit 3 months or 6 months also. The moment we submit and going review process will be take 1 year. So they will lost in between available stability data.
Okay.
As a query, we have to submit it.
Okay, minimum requirement is 6 months.
Minimum 6 months required for the filing part of view, because at least three time points required to be there. So one is 0 month, 3rd month, 6th month.
Okay, like I have one doubt here. See, file IND, when after IND approvals only we will be going for the preclinical studies, right?
No, IND is investigational new drug, which is nothing but clinical studies. So it is a preclinical studies. So preclinical will be is a part of local MOH. It will be not clinical point of view in the respect to MOH. When you want to go for US, we should complete our preclinical studies with respect to Indian market and Indian guidelines, not market, Indian guidelines.
Okay, like for that, what is the approval required?
We have to local MHS to take care. Our Indian regulatory authorities has to be take care.
Local MHS.
Like RCGM and IBSC will be looking to this.
Okay, RCGM. See, like for that, what is the application what is the file will give, like maybe request and file, something like that there will be.
Yeah, we'll have this is one day it's very tough to understand, Raja. So we have a systematic approach. Okay, so initially let's focus on the technical point of view. Okay. And when you come into this part, so we'll come to the applications, everything. We have here a complete package where is shown.
Okay.
Yeah. Where we discussed about PCT application process for the RCGM with reference to the M4 safety guidelines and module 4 complete overview. Okay.
Okay.
It will come systematic way. So one day we dump everything. It may be mess up and it will be difficult to understand.
Okay. Okay.
Yeah. So that's how that is how we required 90 hours time point of view. You have to invest your 90 hours. You will get it. You will become as a master. You will become as a complete knowledge on the biological process, biosimilars, vaccines.
Okay.
Yeah.
Like
so any question? Please.
Bioequivalence will be.
That is for generic medicines,
right? Not for the biosimilars, I feel.
No.
Okay, what is the guideline by the way, like for bioequivalence?
Generic medicines.
Yes.
Yeah, we'll share it that one. Okay.
Okay, thank you.
Yeah. So with the session, Raja.
Yeah, good. I like it.
Okay, fine. So, AK, do you have any questions?
No, thank you.
Is it fine, the session? Okay, fine. Yeah. Ashish?
Yeah.
Any questions?
No.
Okay, fine. Yeah.
Thank you.
Yeah. Abhishek? Abhishek, are you there?
Yeah. No questions. Yeah, I'm
Okay.
there.
Okay, fine. Abhishek More and Abhishek both are same or different?
Both are same, just I have connected the laptop.
Okay, fine. No issue. Yeah. Babji?
I don't have any questions. It was nice session.
Okay, fine. Debarsi? Felix?
No, I'm okay. I don't have any question.
So, are you able to understand the complete picture what we given introduction? Felix?
Yes. Yeah.
Okay.
I understand. I was able to follow everything.
Okay.
My only issue was on the interchangeability, which I think I would have to read a bit before I come back. Thank you.
Yeah, thank you. Yeah, Gladys?
I'm okay. I don't have any question for now.
Okay, yeah, fine, thank you. Yeah, Jagadev?
Yeah. No question. Which is one?
Okay. Okay, so are we clear, Jagadev, the session point of view?
Yes.
Okay, merci.
Thank you for the presentation. And I have no question.
Voice is very low. Otherwise, you drop a text message, I will reply you. Okay. Yeah, Naveen?
Yeah, so I just want to understand. So if we have to get a interchangeable
Concept.
biosimilar provision, you said we have to additionally some switching studies.
Right.
So what exactly we will do in the switching studies?
So in the presentation, I given example here. Let me open that. Yeah, just wait for a while. Others will ask the questions, then we'll come back. Is it okay?
Yeah.
Yeah, Pratiksha?
No, sorry, it's fine.
Okay. And Rajkumar?
None at the moment, but I might have something coming up. I'll connect
Okay.
with you. Yeah.
Sure.
Thanks for the
Satish?
session. It was clear.
Yeah. Satish?
Everything is good. Just to get used to this RA terminologies, if we can, slow down a bit once we pick up, because this may be suitable for who are already in the RA field. People
Yeah, fine.
from
See.
R&D and manufacturing. That is my request.
Sure. See, when you initially when you see the numberings, it will be looks like very tough. But when you go to day by day learning, so it will be become very fingertips. So today is an introduction part. I want to show the guidelines, so I shown that guideline. But when you go to the development point of view, or talking about technical sessions, we'll open particular guideline, particular section. What is the expectation there? What is the ICS guideline expectation? Against that, we'll show the presentation. Then we'll explain the guideline. Then it will be fit into the particular section of the regulatory. Okay.
Excuse me, on the same topic, I'd like just like to add for starters, the few short forms we are not getting, but I'm not getting into it. So for that, you need to explain explain in the
Okay.
French language.
We'll expand it. Okay.
Yeah.
Yeah, fine. Is it clear, Satish?
Yeah. Thank you.
Okay, Sharath?
Yeah, it's very nice session. Thank you. No question.
Okay, any questions, Sharath?
No. As of now.
Are we inline, Sharath, currently developing in the biosimilars?
Yes.
Okay, fine. You can add up anything based on your experience as well. Yeah, all can be add up your experience also.
Sure.
So that will be sharing and sharing knowledge. Yeah, Sweta.
I have a question on interchangeable the products. Maybe I will be waiting for others.
Yeah, no worry. Yeah, it will come back. So what are the shown today overview? We are going to discuss each and every topic. Each and every topic we are going to take up one session separately. Okay. And coming to your question also will be clarified when Naveen question is going to clarify. Yeah. Yes, Naveen, you are asking about interchangeable what additionally we are doing, right?
Yes.
So here I explain. Are you able to see the slide?
Yes.
See, when you're talking about switching studies, when you want to claim as an interchangeable biosimilar, when you file which is the product as identified as a biosimilar already approved or maybe ongoing. Additionally, we are going to generate the data, which is called a switching study. So required for interchangeability proving. So here study design, multiple switch study, at least 3 switches between reference versus biosimilar. So 3 times we have to conduct eliminating the reference product and perform the our interchangeable product, and we have to prove it has giving efficacy and comparative. So we have to do comparative study also. Switching group, reference versus biosimilar, and non-switching group, reference only. So the study especially focusing on PKPD, pharmacodynamics in the clinical point of view endpoints, and immunogenicity, response, and safety. So this is how we have to generate. Additionally, even though these studies will be there, but switching study will be not there in the biosimilars. It is a additional requirement to claim the product as a interchangeable. So when you're having this data, as per the expectation of FDA, when you generate, we'll go for filing. So by generating this, FDA can't approve directly. They will go for review, They will compare it. They will go in deeply of review process. They will raise the queries, and we have to justify with against any queries the data which is insufficient or maybe sufficient, whatever the cases, we have to justify. Then all queries has been addressed and has been satisfied by regulatory bodies. Then only we'll get the approvals. Got it?
Okay.
Yeah.
Yeah.
Yeah. So that is our today's session. We'll connect with tomorrow, starting with cell banks tutoring system. So as per the schedule, will be run Monday to Friday. So you people will block your calendars, and completion of this session, we're going to share the recordings. That is how I'm requesting mail IDs. So we'll share in the same mail IDs the recordings, PPT. So you can use for your, we will give you the access for the download as well as the watch access. You try to download it so that it will be available for you in the lifetime in your systems, right? Yeah. So anyone want to ask any questions further?
No, I have one question. Yeah, related to clinical studies. Okay, so whatever maybe one batch or maybe as per the requirement, we'll go for clinical studies, right? How what is the main role of regulatory from here? So simply getting data or means how much?
Not simply. See, first thing when you're talking about a biological drug or talking about biosimilar drug or when you're talking about any vaccine. So when you open the clinical trials guidelines,
yeah.
So will tell us what to do. So
Okay.
so we'll take up scientific advice. We'll take up. Are able to see the screen?
Yeah. I'm seeing.
Once preclinical is complete, we got the recommendation from agency to carry out clinical. The batches GMP, it should be under GMP control.
Okay.
So once we done GMP, the batches should be made from GMP and testing should be done in the validated condition of methods. So this is, and quality pre-approved specification should be complies.
Okay. So any number of patients will be differ or is there anything?
Yes, it will come. It will come. So during our session, there is a one overview session on the clinical trial. So there is a phase 1, this many people is required. Phase 2, if applicable, this many is required. If phase 3, this many numbers is required. And phase 4, after approval, phase 4, how many patients has to, how many data has to be collected, how many years has to collect, that also will be there.
Okay. Thank you.
Yeah, here the requirement is you people has to be invest 90 hours. You will get it done whatever you want.
Okay.
Yeah.
Thank you.
Yeah, fine. Yeah.